My advice is on how to read chapter 7. So this is known as transcription, to be able to deep read this chapter, scour the figures if they think they already know this topic. So most people have a good understanding of transcription from high school. A great way to start would be to narrate this figure yourselves. So here we have something that you're familiar with already. It has now more detail. So way back in chapter 2, we learned that DNA codes for proteins indirectly, and it has to go through RNA. So we call that the central dogma. Now, this figure has been updated to include additional information. And the additional information here is the direction of the DNA strands, and also the direction of the messenger RNA, also the direction of the protein. So as always, the 5' end of the RNA is coded first, the 3' end is made last. Likewise, the amino terminal of the amino acids is added first, and the carboxyl group at the end is added last. So everything has an orientation. So that's how this diagram has been updated. The question then becomes, which of the 2 strands of DNA is read into the messenger RNA? Next diagram is 7.2. So without reading the legend, students are encouraged to annotate this diagram themselves. So how would you interpret this diagram? Simply by looking at it the first time. It seems that there are 2 genes present on the DNA, and those genes are manufacturing different amounts, different quantities of RNA through the process of transcription, which will be the subject of this segment of this chapter. Subsequently, the RNA, the messenger RNA in blue, is converted to a set of proteins. And all the proteins will be more or less identical, coming from the messenger RNA for that particular gene. So this figure is simply suggesting that genes can be active to different degrees. So gene B is quieter in its transcription activity than gene A. Gene A is producing more transcript than gene B. So this is something that you should be familiar with going forward. Now, even though a gene may be turned on, it may not be turned on to the same degree in every case. So we're left with this scenario. And the way I like to explain this is, in the past, students thought of genes as being on-off, like a light switch. Either the light is 100% on or the light is 100% off. That analogy now has to be modified and replaced with a dimmer switch. A dimmer switch is a better analogy for gene activity. Just like a real dimmer switch, the light can be turned off, or it can be turned up 5%, 10%, all the way up to 100%. And the same thing applies to genes. They can be activated to different degrees. And that's exactly what this figure is trying to convey. This is panel C. We have an RNA molecule. And the RNA molecule seems to have a very similar structure to a DNA. And indeed it does. It has a sugar phosphate backbone, and the phosphodiester bonds are exactly the same. The only difference is that the sugar that's used here is ribose instead of deoxyribose. The other difference is that the base uracil is used in place of thymine. But what we want the students to understand is the difference between thymine and uracil, and that is given in panel B. So here's the structure of uracil, which you don't really need to memorize, but it's nice to know how similar the 2 are. So if you look carefully, everything is the same except at about 10 o'clock on this molecule. At 10 o'clock in uracil, we have just a single hydrogen on that carbon atom, whereas in thymine, we have a methyl group. So this slight change in structure is the only difference that we find. The bottom part here simply shows you how uracil hydrogen bonds across a double-stranded molecule with adenine. Now, you may be asking and saying to yourself, you didn't think that RNA formed double-stranded structures. Well, in the next couple of figures, we'll see that it can in a limited set of circumstances. It can form double-stranded molecules within itself or between itself and a neighboring molecule. So once again, just to conclude, figure 7.3 wants us to realize the differences between DNA and RNA, and the students should be able to recite those if they understand this figure. Going forward, figure 7.5 now gives us additional facts that we may not know. Here we see that RNA molecules can form internal secondary structure. So here's a single RNA chain. It has a 5' end and a 3' end. And when it's released after it's being manufactured from the DNA in the nucleus or even into the cytoplasm of a bacterial cell, it will adopt under those physiological conditions a particular shape. Now, panel A is showing us the conventional shape that students think that these molecules should adopt based on regular base pairing between G's and C's, A's and U's, etc., etc. So these are double-stranded RNA. There's not much of it, but there is significant areas that it does form this man-like structure, a walking man, as we call it. Now, I've highlighted a couple of areas in this figure legend that could be the basis of some in-lecture quizzes. So it says that unconventional base pairs can also form within RNA, like an AG, which is alien to us. We've never seen an AG base pair in the past, but they can form under particular circumstances, and we call them non-conventional base pairs. And if you take this structure and you allow it to form non-conventional base pairs, it actually looks like this inside the nucleus of a cell. So you can see in green, we have those unconventional base pairs, which are permitted by nature under physiological conditions. So the final structure of this RNA molecule would not be as predicted from the knowledge that we have so far. Like in panel A, it would be more like in panel B. So panel C actually shows us the structure of the RNA molecule. It actually adopts. And this molecule here is a very special molecule because it does perform RNA splicing, right? So we'll come back and look at that later. But you can see this single figure has a lot more information than we thought we would decipher based on what we know so far. So here's added information coming out just from the figure legend. So when you're reading this, you should be also thinking about that. So first thing, most important, is that you learn what the information is. Don't memorize it, but learn it. And then come back again, a second glance, and have a look and ask yourselves, what would the instructors think is relevant for asking questions from here? So let's move on to figure 7.6. Figure 7.6 just goes on to help us understand, if I go back slightly, please, it helps us understand the question that we asked earlier. Which of these two strands of DNA is used for making the RNA? Are they both used? Or is the top one used every time? Or the bottom one? Or is there a convention? And now this figure is going to tell us that only one strand of the DNA is used every time for making the RNA for that particular gene. And we have to emphasize that, for that particular gene, right? So in this case, the bottom strand will be used. How do I know that? Because it tells us in the legend that the template strand is the one that is read by the enzyme RNA polymerase, the enzyme that copies DNA into RNA. If you look very carefully, the color coding is complementary to the bottom strand. So there's a green nucleotide there, and the RNA puts in a red nucleotide. There's a yellow nucleotide here, and the RNA puts in a blue nucleotide, etc., etc. So the bottom strand is used for making RNA. So what about the top strand? Well, the top strand is not necessary in this case. It's only there because of the need to copy DNA, etc., etc., that we learned in chapter 6. So that is called the coding strand, which can be confusing. But the two terms come from different branches of science. The coding strand. Now, if some of you have a good eye, you will notice that if you look carefully at the color code of the messenger RNA, they correspond exactly with the color coding of the non-template strand, the top strand. So red here, red there, blue there, blue there, yellow, 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 yellow. So that's why a geneticist called the top strand the coding strand, because it carries exactly the same information as the messenger RNA, or the RNA in this case, that's made from that gene. Except that wherever there's a T in DNA, we find a U in RNA. Everything we learned in chapter 6 has to apply here. So DNA is read in one direction, read, and then it's made in a different direction. So let's have a look. We learned in the past that DNA is made backwards. So DNA is made backwards, so is RNA. RNA is made backwards too. But this part of the molecule here is the 5' end. This part of the molecule is the 3' end. So if we go backwards 5, 4, 3, this tells us that this side of the molecule was made first. This side of the molecule was made recently last. So this follows the same convention as DNA. So if things are made backwards, then in which direction is the template strand read? The reading and making are different things. So the template strand must have been read forwards. So the bottom strand was read 3, 4, 5 in the forward direction in order to make the RNA strand backwards. That applies to all nucleic acids, not just DNA. Figure 7.7 actually shows you and reaffirms what we just said on figure 7.6, that one strand is read forwards in order to make something backwards. So the bottom strand is going in the 3, 4, 5 direction, so therefore it must be read in the forward direction, 3, 4, 5. And this enzyme in blue, light blue, is traveling along the bottom strand, only reading that information. The top strand is just displaced to the side. It's not read, it doesn't do any other function except to act as double-stranded DNA. So the messenger RNA in this case, or the RNA, because I keep saying messenger RNA, this could be any type of RNA. This could be ribosomal RNA, this could be transfer RNA, or it could be messenger RNA, the most common types of RNA. But all RNA is made in this exact same mechanism. So nucleotides are brought in by RNA polymerase, not DNA polymerase in this case, and they're polymerized into a chain which corresponds almost exactly to the sequence of the information carried in the coding strand on top. So once you've explained that to yourself or your colleagues, then it's a good idea always to then read the corresponding legend that's been written by the authors of the textbook. And what I like to do is highlight within that the most important concepts that have been brought to my attention, something new that I didn't know earlier. So I would highlight certain regions of this, maybe 4 or 5 regions of this legend to help me remember those particular components, right? So there's a DNA hybrid, RNA hybrid, that's important to understand. It goes on to reaffirm the direction in which material is utilized and read. So this is the reading direction, not the making direction, right? And then it talks about how the polymerase, the RNA polymerase, moves along the DNA. Figure 7.8 is simply there as a witness to show us that we have observed this process in action under the electron microscope. Now, the reason we use the electron microscope is because the light microscope isn't powerful enough. So I would highlight that this picture was taken by an electron microscope and not a light microscope, just for the sake of adding to what we learned in chapter 1 about size and scale and everything else. So this scale is 1 micrometer, right? So you can see here, the very thin line, I guess, would be DNA. So, because I'm not reading this yet, I'm just explaining this to myself. So I guess that the thin line will be DNA, and on the DNA, maybe these little Christmas tree-like structures that are appearing seem to be that whatever happens starts at the beginning, and as it moves down along the DNA, it produces something longer and longer. And the same thing happens in the neighboring gene. So what are these things? Well, if that is DNA in my first interpretation, then this could be RNA coming out to the side. And the question is, am I right? So then I would read the figure by uncovering it and read the legend to the figure by uncovering it and see how much of that information I got right and how much of that information I got wrong. So that's the first thing I would do. The second thing I would do is read that again afterwards once I understand it. So let's quickly read it together. So it says the micrograph. And what is a micrograph? Well, that's the thing. That's something new that you may not know. A micrograph is a picture taken by an electron microscope, not a regular light microscope. So that's an important terminology that you may not have known. So the micrograph shows many molecules of RNA polymerase simultaneously transcribing two adjacent ribosomal genes on a single DNA molecule. So our understanding may have been right, that there's one gene here and there's another gene here next to it. What type of genes are these? Well, these are ribosomal RNA genes, right? So these are the genes that make ribosomal proteins, proteins that are part of the ribosome, which we'll reconfirm later on. So molecules of RNA polymerase are barely visible as a series of tiny dots along the spine of the DNA molecule. So these little tiny dots that you see here, let me get my cursor going. These little tiny dots that you see on the DNA are the RNA polymerase. So the RNA polymerase can be seen under the electron microscope, right? And each polymerase has an RNA transcript, a short fine thread that radiates from it. So these little threads coming out is RNA. So it comes out almost at 90 degrees from the DNA molecule. The RNA molecules are being transcribed from the two ribosomal genes, ribosomal RNAs, are not translated into protein. So these RNAs, these RNA molecules, will not be then sent to the ribosome. They are now finished. They will be then used as is to construct the ribosomes. So that's what it's saying here. So we can highlight that by saying not all RNA is made into protein. So that's the information that I would learn simply by looking at that figure. So then it goes on to say the large particles that can be seen at the free 5' end of each RNA transcript. Where is that? Well, if we go back to the figure above, the 5' end of a messenger RNA or any RNA is produced first. And then that drifts into space as the RNA polymerase copies further and further. So it's likely that they're talking about these ends here, the extreme ends right there, where the little dots are, okay? So that's what this part of the figure is saying. The large particles that can be seen at the free 5' end of each RNA transcript are believed to be ribosomal proteins that have assembled on the ends of the growing transcripts, right? So it looks like proteins are coming in, and as soon as the RNA molecule is made, the proteins, which are different, grab onto those RNAs and start forming a blended structure. So think about it. What is a ribosome? Ribosome consists of a small subunit and a large subunit. You should know this from high school. And within each of those subunits, the large or the small, there's a large complement of protein and a significant component of RNA. Well, that's what's going on here. The two things are blending together as soon as the RNA is made. So this happens in the nucleolus of a typical eukaryotic cell or in the cytoplasm of a prokaryotic cell. So you see how I'm using information that we learned in the past to assemble a bigger picture of how things are going on. So that's what I like students to do more and more. It's a skill that you must learn. It's a skill that will help you succeed in the future. So once again, a simple drawing that some students in the past would have skipped over. It's a great place to revise, to relate to different things, and to just build your knowledge further. The different types of RNA that can be produced by cells. So we're all familiar with messenger RNA, and indeed it's the number one on that list. So messenger RNA, its function is to take information to the ribosomes and code for proteins. So all of us should know this from high school. We should also know about ribosomal RNAs, and they are the structural components of ribosomes. They construct the ribosome. Then we have transfer RNA, let's miss one. Let's go to the fourth box. Transfer RNAs, they exist in all living things, and they're the bridge between amino acids and messenger RNA. So transfer RNAs bring to the ribosome the amino acids that are necessary to construct the polypeptides. And that's going to be the subject of the middle part of this chapter. So we can leave it until then, but now we have a good understanding of what transfer RNAs do. They deliver, using special chemical bonds, the amino acids to the ribosome. But there are two other lines here that add to our knowledge. It says, in particular, that microRNAs, miRNAs, microRNAs have been discovered in the last 20 years, and they, believe it or not, they control, that's what the word regulate can be translated to, they control gene expression. They control how genes are activated and by how much. So this is a more recent finding. So these tiny little RNAs, that's why they're called micro, because they're small in length. They interact with in the nucleus to control the activity of which genes are turned on and off, which suggests that RNA has a bigger role than we gave it in the past. And at the bottom, we have a general compartment for all the other types of RNA. So if you were just reading this book, you would think, okay, there's 5 types of RNA, and that's it. But if you go to Wikipedia, and it takes a few minutes, and you were to check there on the number of types of RNA that are available and have been recognized to date, you will see a list approaching 20 different types. So this table could be replaced in the next edition of the book with a bigger table that lists 20 different types, in addition to these others, different types of RNA. So we're discovering more and more RNAs than we ever discovered in the past. Okay, so they all lump together at the end of this table. So that's important. So this table is adding to our knowledge, and tables are normally structural, they're foundational. You mustn't ignore them. They must become the bedrock of your understanding. So they contain important information. So earlier on, back here, they were just telling us that genes are transcribed from the DNA, but where does it start? Where's the beginning of the gene? Where's the end of the gene? We see down here that there's a beginning of a gene and there's an ending of a gene, because everything stops at that point. It doesn't continue. It doesn't continue to get longer and longer. So each gene must have a beginning and an end. And what this figure here is doing, figure 7.6, is giving us further information to confirm that we have now the understanding to notice that each gene has a beginning called the start site and the end called the stop site. So every gene known has to have these two components. The start site is where the beginning of the RNA transcription begins. So the very first base put into RNA from the DNA template strand is made at the start. And the stop site is the very last nucleotide that's incorporated into the RNA molecule. So these two must be very important locations on a gene. How do we know that? From experiments. If the start site has some type of mutation, so the sequence changes, then the beginning of the gene cannot be activated. If we mutate through similar events by changing the nucleotide sequence of the DNA, we mutate the stop site, then sometimes the RNA polymerase doesn't stop there. It keeps going on and on and on until it hits another natural stop site, which may be a long distance downstream. Well, let's get back to this figure, right? So this figure is one of my favorites in the book because it uses nice colors to illustrate something important going on. Now, they do tell you straight away that this is bacterial RNA polymerase. So I would highlight that. So the reason they put the word bacterial there must be because eukaryotes and other creatures must do it differently. Yes, and that's really is the case. So you're looking at subsection of transcription, only applying to eukaryotes. Sorry, wrong word. Only applying to prokaryotes, bacteria. So bacteria must have this structure, and maybe eukaryotes have something different. We'll have to wait until subsequent figures. But for now, let's apply this to our understanding of prokaryotes. So in prokaryotes, there is a start site, there's a stop site on either side of the gene, and there's something called a promoter site. So the promoter site exists, that's in green here, that exists at the beginning or around the beginning of every gene. And that's to which the RNA polymerase will bind. The question is, can the RNA polymerase bind itself, or does it need the help of another protein? If you read the legend, which we'll do in a second, it seems that it's a cooperative binding. So that yellow molecule there that you see must be necessary in order to bind to the promoter first, and then it calls in the RNA polymerase to the start site. And it appears that once you start transcription, the yellow molecule falls out. Yes, so the yellow molecule called sigma is now no longer necessary. So I would highlight sigma as being a very important component of our understanding. So initiation is begun by the sigma factor, and then once the sigma factor has done its job, it looks like transcription continues without sigma until the stop site is reached. And at the stop site, it looks like the RNA polymerase falls off and reassociates back with sigma to maybe go back to the beginning of this gene or any other gene. But the transcript is released. So this is called a transcript because it's produced by transcription. And it has the 5' end, which is the side that was made first. And then it has a 3' end, the side that was made last. You'll notice that I'm repeating myself again and again because that's a necessary component of learning and revision. So everything I learned back here, when I was talking about this system, right, with RNA being made, etc., and I spoke about the orientation of the nucleic acids, I'm reinforcing by speaking again and again. I spoke about the 3' end and the 5' end down here. And I spoke about the 3' end and the 5' end back here again. And now I'm speaking about the same components here. So at least 4 or 5 times during the last 40 minutes, I have repeated myself multiple times whilst going through this chapter. Now, naturally, I would go faster than this because I then have to speak to the microphone. So during my regular reading, I would be here in about 10, 15 minutes, right? So that's what I expect you to pick up the pace whenever you're doing your own personal studying. We don't have too much information about the stop site. There's also something called a terminator here that we haven't really spoken much about. So let's read the legend and see if that can add to our knowledge. So I'll read it very quickly. Signals in the nucleotide sequence of a gene tell bacterial RNA polymerase where to start and stop transcription. So these are called signals. And those signals are not carried by the RNA polymerase. They're not carried by the RNA. They're actually encoded in the DNA itself. So DNA is a wonderful molecule that can carry lots of information. Not only does it carry the information for making the RNA, it also carries information for telling the system where to begin and where to stop copying that particular gene. So reading through into the legend, bacterial RNA polymerase, light blue, contains a subunit called sigma factor. The word factor means protein, right? So let's highlight that. That's a protein. So the yellow molecule is a protein. Called a sigma factor that recognizes the promoter of the gene. So the sigma factor recognizes the promoter, not the RNA polymerase. So that could be a question for an in-lecture quiz or even a homework, right? So once transcription has begun, sigma factor is released. It's no longer necessary. So it's only necessary for binding to the promoter and getting transcription started. So reading further, and the polymerase moves forward and continues synthesizing the RNA without the sigma factor. So chain elongation, what the hell is that? So let's highlight that because that is something the TAs may pick up on. So chain elongation continues until the polymerase encounters a sequence in the DNA called the terminator. All right. So the terminator is a sequence on the DNA at the end of the gene, which is important. So the TAs could ask you, what is a terminator? There, the enzyme halts and releases both the DNA template and the newly made RNA transcript. So when this light blue molecule, the enzyme, gets to the terminator, it will then fall off the DNA, releasing the DNA, and also at the same time, releasing into the environment the transcript, the RNA molecule. Okay. The polymerase then reassociates, okay, let's highlight that. That means it reconnects with this free sigma factor and searches for another promoter to begin the process again. So as we thought earlier, the two unite and they go looking for another promoter that needs their help. So that's the way we can interpret that figure. So naturally, the next figure, 710, says, okay, folks, let's have a look at both those very important elements on the DNA. So let's look at the start signal in more detail and the stop signal. So the start signal is called the promoter. The stop signal is part of the terminator. That's what it looks like. Look, there's the stop signal and there's the start signal, but it seems to be part of a bigger entity. So the start and the stop signal are parts of something called the promoter and the terminator, right? So if I were to ask you a question in the future, where do you find the start site? You could say the start site is part of a promoter. And if I say, where is the stop site? You could say that the stop site could be a part of a terminator. And you will be right in that analysis. So here we see that we have double-stranded DNA. The bottom strand is the strand that's being read by the RNA polymerase. But this is where the RNA polymerase will attach to separate the two strands. How do I know that? Because if you go back to figures here, you can see that the two strands of DNA appear to be separated, and the bottom strand is the one that's being used to make the transcript. So that bubble, that tiny little bubble that exists inside the RNA polymerase must be made somewhere. And it's made here at the promoter. What does this suggest? Well, the +1 seems to suggest something. The -10 seems to suggest something. The -35 seems to suggest something. The green boxes suggest something else. Oh, and there's a bit more information here. Look, we have the RNA at the 5' end. The A corresponds to maybe the A here. Yes, so that A is similar to this A. Then we have two G's and a U, two G's and a U, because DNA doesn't contain U's, it contains T. So this sequence here corresponds to the sequence there. Very interesting. So it appears that the +1 site at the beginning of every gene is corresponding to the very first nucleotide incorporated into RNA. So that's a very important understanding. Now, I suspect that there's a little error in the drawing here, and that this should be one nucleotide back. The whole thing should be pushed back to line up with this. You see it doesn't line up perfectly? So that's an error by the people that drew the figure. They should go slightly back. But regardless, it does help us. In fact, it does us a favor by doing that, because we now have to interpret what that means. So the conclusion is that the +1 site, whenever you see that in the future, will be the place at which the RNA first nucleotide is added by the DNA polymerase. So what about the rest of it here? Well, if you count backwards, then it must correspond to this. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Huh? So 10 bases back from the point at which RNA is made is labeled as -10. And if you were to count this, I won't do that now, but if you were to count this, you'd find that this T on the top strand is -35 from this position. So these are very important findings that we have come across in the past that tell us that the region upstream, and this is called upstream, like water flowing this way, this region upstream of the start site seems to have a very important role in positioning the RNA polymerase so that it starts copying DNA at the right place. So the promoter can be reclassified as an important region of the DNA which positions the enzyme RNA polymerase to start copying the gene at the correct location. Because you don't want to start copying the gene here or down here, because then the information is going to be incorrect. So this is a very, very, very precise mechanism that cannot afford to miss by one or either side. So now let's look at the other side of the gene, the terminator. So the terminator must be important in telling the enzyme when to stop copying the RNA. So let's follow with this convention. So the RNA started here. The arrow tells us that it keeps on going. And here's the same RNA. You can see it here coming in, right? The other side of it, it could be 1,000 bases long. It doesn't matter. We're now hitting the other side of the gene. So you can see the RNA stops being coded right here. So those 4 T's in this particular case are incorporated into the RNA. Remember, RNA contains U's, not T's. So that reconfirms our understanding previously. But after that last T, we don't see any other nucleotides. So the stop site and the start site correspond to the start and the ending of the RNA molecule made by that gene. Okay? So that's what the stop site is. Now, what are all these boxes that are colored in here? Well, they may be important in controlling when the stop site is activated. What about these green boxes? They may be important in controlling how you start from the +1. So let's read the legend and see if that confirms our understanding that the green boxes and the brown boxes are very important. So let's go. So bacterial, oh no, now we're back to bacteria again. So eukaryotes must be different because the authors would not waste their ink telling us this is bacterial unless it was important. So bacterial promoters and terminators, these 2 regions, have specific nucleotide sequences, specific, okay, let's highlight that word, they have to be particular, nucleotide sequences that are recognized by RNA polymerase. So these sequences must be, the shape of these sequences must be recognized by these enzymes that interact with the DNA, the RNA polymerase, the sigma factor, etc. All right. So I'm going to read section A without stopping until we get down to here, right? So I'm going to read all this. So it says the green shaded regions represent the nucleotide sequences that specify a promoter. Okay, so this region and this region must be the heart of the promoter. I said I would read without stopping, so I'm sorry. The green shaded regions represent the nucleotide sequences that specify a promoter. The numbers above the DNA indicate the positions of nucleotides counting from the first nucleotide transcribed, which is designated +1. The polarity of the promoter orients the polymerase and determines which DNA strand is transcribed. Oh my God. All right. Highlight that. All bacterial promoters contain DNA sequences at -10 and -35 that closely resemble those shown here. All right. Okay. So it seems that this TATATT is present nearly every time that you find a gene in bacteria. And also, this TTGACA is also found upstream of this box, this box. We call these promoter boxes, right? So these boxes are found all the time upstream of a transcription start site. So these DNA sequences in between may not be that important. They just may be important for making sure that the distance between the 2 boxes is maintained. And as you'll find out a bit later, that is exactly the case, right? So the RNA polymerase, let me go back one slide. This light blue molecule, when it sits down on the promoter, is actually making contact with this part of the DNA and this part of the DNA. And it's sliding up and down looking for these bases, right? And the sigma factor is also important in that. So when it finds and makes contact with the inside of the DNA helix at these 2 locations, the front of the molecule is making contact here with the +1 site. If you came in the other way, then the enzyme would not be able to do that. So if you came from the top with your hand coming down, you couldn't put your hand on these elements. But if you came from the bottom, you would be able to make your finger touch this and your thumb touch that and start copying here. So this sequence has a direction. It's facing this way. It's not facing the other way, right? Therefore, when the DNA polymerase comes and sits down on this piece of DNA, it's automatically making contact with that A. And once everything is perfect, then the polymerase separates the DNA here into two strands, the top and the bottom, right? And forms that tiny little bubble, and it starts copying the bottom strand. So this information is very important. This word, the polarity, is key. Polarity simply means direction. So the direction in which the RNA polymerase sits down automatically sends it on the right strand of DNA going in the right direction, which is amazing in itself. So let's finish off this figure quickly. It says in part B, the red shaded regions represent sequences in the gene that signal the RNA polymerase to terminate transcription. Note that the regions transcribed into RNA contain the terminator. By convention, the sequence of a gene is that of the non-template strand. As this strand has the same sequence as the nucleotide. What? So it says the red shaded regions represent the sequences in the gene that signal the RNA polymerase to terminate transcription. So these three boxes here, the ones that are colored in this red brownie color, these regions are very important. So when the polymerase, which is a large molecule, the size of this circle that I'm drawing, when it reaches this region here, it interacts with these DNA nucleotides. And they cause it to stop moving forward. They hold it very tightly, so it stalls. And as it stalls, it then falls off the DNA. So it will automatically always stop right here. And therefore, the last base added into the RNA will be that last U that you see there. Every time. It will never miss it by one, either side. It's very, very precise. Very similar to the way that the promoter works. So these red boxes or brown boxes are very important, right, for termination. And it also goes on to say that that sequence is part of the final RNA, all right? So this sequence here, the G double C, G double C, A, G, will be found inside the actual RNA. What does that mean? Nothing at the moment. So all they're saying is that the promoter sequence is not part of the transcript, but the terminator sequence seems to be. Right? What it actually is. Then it goes on to talk about something else here. It says, note that the regions transcribed in the RNA contain the terminator, but not the promoter sequence. We just said that. By convention, the sequence of the gene is that of the non-template strand. All right? So if I was to publish this DNA sequence in a paper, and I said I found this gene, I would start the gene at this +1. But which of the two strands should I type into my paper? The top strand or the bottom strand? It's the bottom strand that's actually being used to make the RNA. But as it says here, by convention, we scientists have decided that the sequence that most looks like the messenger RNA, i.e. the top strand, is the one that we would publish. So if I wanted to publish this gene sequence, I would start with A, G, G, T, C, C, A, C, G, etc. Right? That's the only sequence I need to publish. I do not need to publish both the strands, nor will I publish the bottom sequence, because that would be then going against convention. So the next figure is at the bottom. And what is it trying to say? Well, don't read the legend yet. Don't read that. Just look at the figure itself. What does it seem to be saying? It seems to be saying that this is a piece of DNA, okay? And there's a gene A on it, which seems to be producing RNA in that direction. There's a gene B on it, which seems to be producing RNA in the opposite direction. How can that be? Well, if we remember what we learned up here, the promoter is always at the beginning of the gene. So if this gene is being transcribed in that direction, the promoter must be here, and there it is. The terminator must be here, okay? And if this gene is being coded in that direction, the promoter must be here, and the terminator must be there. And that seems to be the case. But from what we've learned in the last few diagrams, if the gene is going in this direction, the bottom strand must be carrying the template strand. The bottom strand must be the one that's been actually read by the enzyme and copied into the transcript. And that's true. If the gene is pointing in this direction, then the top strand of the DNA must be being read, not the bottom strand. And it must be read in the forward direction, 3, 4, 5. And look at that, it is read in the forward direction. So the top is the template here, and the bottom is the template here. So that's fascinating, that neighboring genes can either point in the same direction or in the opposite direction. And they're pointing in the opposite direction here. And wherever they're pointing, the promoter is normally found, I should say at this point, is always found at the beginning of the gene. Although, sometimes it's found in the middle of the gene. But that's a different story. So let's read the legend very quickly. So on an individual chromosome, some genes are transcribed using one strand as the template, and other genes are transcribed using the other strand. Yeah. So you had 50 genes here. Some of them would go in the same direction, others would go in the opposite direction. Those are the two choices. RNA polymerase always moves in the RNA polymerase always moves in the forward direction when reading the DNA, and the selection of the template strand is determined by the orientation of the promoter. Aha. So the orientation of the promoter. So going back to the figure above, the -10 and the -35, they will decide which direction the polymerase would be pushed, right? So in this case, when they sit down here, that sequence would push it to the left. How do we know that? Because we can turn this around using experiments. We can turn the promoter around by flipping it. In that case, the RNA polymerase will move to the right. So all this has been tested and proven using experiment after experiment after experiment. So let's finish off reading the legend. So RNA polymerase always moves in the 3 to 5 direction when reading the DNA, and the selection of the template strand is determined by the orientation of the promoter at the beginning of each gene. Thus, the genes transcribed from left to right use the bottom DNA strand as the template, and those transcribed from right to left use the top DNA strand as template.